Yes, sir.
Yeah. Any doubts? Anything you want to ask? No doubts? If there are no doubts, then someone has to quickly summarize what we did in the last class. Where did we start in the last class? I kind of microbes in sewage treatment. Oh, I think I started from treatment and therapies, right? From streptokinase. Okay, anyone who would like to give a quick summary? Anyone who would like to volunteer? See, you should volunteer by now. Don't make me cherry pick every time. No one would like to volunteer? See, if we cannot get away with summaries this way, then I'll pick someone randomly. Okay, how about Minashi? Minashi, you're there?
Sir, I couldn't join last class.
Okay. Did you go back and
No, sir. I was having exam.
you were having exam? Okay. So about Mamuna. Mamuna, you were there in the last class? Mamuna, would you like to summarize? It's just a good practice for you. I'll be there. So if you get stuck somewhere, I'll just help you. In fact, it's we both are summarizing together kind of thing. I think Mamuna cannot hear me or cannot speak from her behalf. So I'm left with Samira.
So this is my first class in this batch. I don't know which chapter is this.
Okay. So we are actually doing. Okay, Samira, I'm so sorry. I just forgot. We are doing chapter 10, the good microbes in human welfare. Okay. And after this, I think you joined this for biotechnology,
Yes.
right? Which we'll be doing tomorrow.
Okay. I was told that it was going to be done today. It's going to be started today.
We still have to finish biocontrol agents. I'll try if we can start some part of it today. That will be good. But you can also join tomorrow because we'll do that tomorrow. So we are doing microbes in human welfare. And I think we have someone joined. Saifullah joined. Saifullah, how are you?
I'm fine, sir. How are you, sir?
Yes, we were looking for someone who can rescue us and help us with some with the summary. And then you joined. Would you like to summarize the last class?
Yeah, okay, sir.
Yes, thanks. Okay, so Samira, after the summary, we'll do the remaining part of this chapter and we'll see if we can start biotechnology today. Okay.
Okay, sir.
Yeah. Okay. Saifullah, over to you.
So,
Summarize from streptococcus, like
yes.
streptokinase. Yes.
There is a medicine called streptokinase, which is derived from a bacteria known as Streptococcus. After some genetic modification, it is used as clotbusters for patients of a myocardial infraction.
Yeah, myocardial infarction, FAR.
Yes. It
leads to heart attack.
Yes, it
Yeah.
leads to heart attacks. Then it is cyclosporin A. It is a immunosuppressant agent. It suppresses the immune system of a patient with organ transplant. It is derived from a fungus known as Trichoderma polysporum.
Yes.
It is a bioactive molecule.
Yes, all of these are bioactive molecules. Bioactive means they are active in the bio in the living system.
Yes. Then it is statin. It is a medicine used for lowering blood cholesterol levels.
Yes.
It is It is produced from a fungus known as Monoscus purpurus.
Purpurus, yes.
Then we went to sewage treatment. Like sewage is the wastewater that is secreted from our houses every day. It contains lot of pathogens, harmful pathogens, human excreta, and other toxic materials.
What is the major component of a sewage?
Human excreta.
Yes, right. So that's why we need to treat it. Why do we need to treat it basically? Because
So if we like directly pour it into a water body, it will contaminate the water body, make it as harmful and for use and the aquatic life for drinking purposes also.
yes, the aquatic life will also suffer and the water will not be fit for drinking purposes. And basically we can lead to a widespread collapse and also diseases can spread. So we have to treat it. Okay, cool. So how are we going to do that?
It is treated by two stages. First is primary stage, and the second one is secondary stage or biological stage, biological treatment.
Okay.
In primary treatment, it's the simple filtration takes place in which plastics or cans are removed by a belt that secretes it by filtration.
Yeah, so there are two things in primary treatment, Saifullah. It's simply the physical removal of substances. Now there can be two type of substances. One is that float and the other is that sediment, right?
Yes.
So that could be plastic bottles, polythenes. It could also be the human excreta, could also be hair or whatever kind of waste is present inside it. If it is something which can float, it can be taken out through filtration, the floating waste. If it is something which is heavier and sediments, then it can be taken out after sedimentation. Yeah. Yes, continue, please.
Yes. And then this after filtration, it goes to like sediment tanks, like sedimentary tanks in which sedimentation takes place, and the materials that settle down is called the primary sludge, and the water that is present, it's the effluent.
Yes, effluent is when you remove the floating debris, you also remove the sedimented debris. Whatever is left is not the effluent. And this is the part which we want to treat, right? Because this is the water that we want to throw into the rivers later on.
Yes.
Yep.
This effluent is then processed in secondary treatment, also known as biological treatments.
Mhm.
The effluent is passed through a aeration tank in which it is constantly agitated and water and air is continuously pumped into it
Why?
for the growth of aerobic bacteria.
Okay. Why do you want to grow aerobic bacteria?
Because they have to digest the anaerobic organic matters.
Yes, they will not digest the anaerobic bacteria per se, but the anaerobic bacteria will automatically start dying when we will put oxygen, because they will not be able to tolerate oxygen levels, right? But that water, that wastewater also has a lot of organic matter. That organic matter is actually the problem, the toxic substance that we want to deal with. That's why we are trying to grow aerobic bacteria with the help of aeration, so that they can consume all that organic substance and break it down into simple and harmless chemicals. Okay. Yes, continue then.
There is a term called BOD. BOD means biochemical oxygen demand,
Yes.
which refers to the amount of oxygen consumed by the organic substance.
Mhm.
This indirectly gives us the measurement for the bacteria that are present in it. If the BOD is high, that means the polluting potential is also high. If it is low, the polluting potential is low.
So
It is
a polluted water will have high BOD or low BOD?
high,
High BOD. Okay. And a pure water from a glacier will have
low.
low. Okay, cool. Yes, continue then.
Um, and then after agitation, the effluent is passed through like sedimentation tank in which yeah, it is again sedimentated, and now the sediments that we get is known as activated sludge
Why is that?
because it has some bacteria, sir. Those
It has
bacteria.
aerobic bacteria. Anaerobic so we killed no already when we put air. This has aerobic bacteria and that is called activated sludge because this has the bacteria that we need to grow. So some part of it can go directly into the first process, the first step. And the most of this remaining part of this activated sludge goes into anaerobic sludge digester, where anaerobic bacteria kill these aerobic bacteria and digest it. Correct?
Yes, sir.
And in the process, methane, carbon dioxide, and hydrogen disulfide is released, and we get the biogas, right?
Yes, sir.
Which is a fuel source. And what happened to that effluent when we got the activated sludge that settled down? What happened to the secondary effluent?
So it is now released to the water bodies.
Yes, because it is safe to be released. Correct?
Yes.
Okay, very well. Thank you so much, Saifullah. So now let me begin with, so we already know that we also have microbes that apart from playing roles in sewage treatment, microbes which help in production of biogas, right? So I told you about biogas. But it's not like all the time we make biogas from the sewer treatment plants. We can also make biogas separately just by setting up biogas production plants. But one concept will stay the same. What do we need? We need bacteria that can produce methane, right? Because what is a biogas? Biogas is predominantly methane. It's a mixture of, it has hydrogen disulfide, it has CO2, but predominantly what it has?
Methane.
Methane. So we need bacteria that produce methane, and these bacteria are collectively called methanogens. Okay. Biogas is produced by the action of methanogens. And if you ask what is a methanogen, then methanogens, a bacteria that produces methane. Okay, everyone? Is it clear?
So,
Yeah. Now, one such bacteria that produces methanogen is called methanobacterium. Simple. Good name. So example is methanobacterium. Any other you know about? Any other common, very common bacteria? Okay, where do you think these methanogens are found? Where do?
in cows, buffaloes, gut.
Oh yes, so they are also found in, apart from, I was expecting an answer that these methanobacterium will be found in this anaerobic sludge digester. Okay, because that's where this methane is coming from, correct?
Yes.
So, and apart from the anaerobic sludge digester, these methanogens are also found, as FLS correctly pointed out, in the where?
Gut of cows, buffaloes.
Gut. Okay, yes, you use the word gut. That's correct. But where in the gut? Everywhere.
In the rumen type.
Yes. What is rumen? What is a rumen? Anyone?
So it's a part of a stomach of cows, buffaloes.
Yes. Found in the rumen of cattle. Let's say cows and buffaloes. You know what are cows, buffaloes, and these cattle called? They are called ruminants. You know that? Ruminants, because they have rumen. Rumen is a okay. How many of you have seen cattle's grazing? Anyone who has seen cattle's grazing? Reem has also joined us. Good evening, Reem. How are you?
Good evening, sir. I'm good. How are you?
I'm great. I'm great. So, have you seen cattle's grazing even in movies?
Maybe, sir.
Yeah. So, how do they graze?
They use few instruments.
They use
Few instruments.
instruments. What instrument? Cattle's, like cows and buffaloes. How do they graze the grasses? What instruments are cattle going to use? I'm sure that you got confused with some other question, or you misunderstood the question. I'm simply asking how do cows and buffaloes eat grass? So if you see them, when they eat in the fields, they're just cutting the grass and just swallowing it. They're not chewing it. They never chew the grass when they're in the fields. You just cut it, and then just gulp it, swallow it. And all that grass goes, so rumen is an additional stomach that these ruminants have, in which there are methanogens. So these methanogens ferment and produce lots and lots of methane, and also act on the starch or the carbohydrate component. After some time, when these cattle sit, you must have seen cattle always are moving their jaws when they're sitting, as if they are chewing the chewing gum. You know, so if you are constantly chewing the gum, sometimes teachers or parents often say that, why are you grazing like a cattle all the time? Yes, no, never heard of it. Please say something.
Yes, sir.
Yeah, so that's called a additional stomach, where the food, before they chew it, gets partially digested by these methanogens. Okay, so they are present, they are bacteria, they are helpful bacteria, and they are present in the rumen, and they help, write down, they help in the breakdown of cellulose. You know, cellulose is very difficult to break. That's why when we eat leafy vegetables, we only absorb the starch from it, not the cellulose. We cannot digest cellulose as humans, because we do not have a rumen. We also do not have methanogens living in our gut. So they help in the breakdown of cellulose and play important role in nutrition of cattle. Okay. Is it clear?
Yes, sir.
Yeah. Now, just because of this region, because they have rumen and they have lots of methanogens in the rumen, which partially digest the food before they digest it properly. But after a proper digestion, when they excrete finally, so that's called cow dung or gobar. So cow dung has a very high number of these bacteria. So the excreta of the cow or the cattle, which is commonly known as the cow dung or the cattle dung, is rich in these methanogen bacteria. Okay. So cow dung is rich in methanogenic bacteria. So what is the benefit? Should I be happy as someone who wants to generate or make biogas? Is it a good thing? Apart from making methane from sewage treatment, can we use cow dung to make methane and biogas?
Yes, sir.
How? Can you think idea for me? What should I do? What should I do? Come on, how will you do it?
So we can let the bacteria digest cellulose and release methane.
Okay, so if you want to produce biogas, how will you? Yes, you are correct. You are very correct. So let's try to see it. So how a typical biogas plant is, where we do not. So here the complexity of a primary effluent, a secondary effluent is not there. So what we need very simple two things. This is the ground, let's say. Okay. First, we need to dig a hole in the ground. What is this hole that we have dug? This hole is to put in dung mixed with water. So we take cow dung, we make a slurry of the cow dung, and we put it in this first tank. So what should I call this tank? Let me call it collector tank. Can I call it collector tank for the dung? Now this tank is joined to a bigger tank, a very big tank actually. So let's say this is the bigger tank. Okay. Now in this bigger tank, the point is you have some slurry, let's say. And it will come and start collecting here. Over time, the level will rise. And what we do is collector tank, we put on top of it. So because this is also a well, so on top of it, we'll put a floating cap. What are you putting? A floating cap. Now this floating cap can just float over the tank, and then later on, this is the outlet of the gas. So what happens? Let's pay attention here. We put dung plus water slurry here because we know that it is rich in methanogens. We'll just keep it undisturbed for many days. Now the number of methanogens will add and methane gas will be produced here. So in this, it's all getting fermented, and we know that lots of methane gas is producing. Now it will go and get trapped into this floating lid. And from that, in this floating lid, we will have methane. So we have hydrogen disulfide. We have CO2. Now all these gases will rise the chamber, and these gases are good to be collected and used for household purposes through in the form of gobar gas. Now this chamber will eventually fill up, right? If we keep adding dung and water here and it's connected to this chamber will fill up over time.
Yes, sir.
Yeah. So what we do is we make another connection here like this. Okay. Now this will be known as a sludge collector. And whatever, when this chamber will start rising and will reach here, it will start flowing here and will come and deposit in this sludge collector. Now we know that this part is not very useful now because it has already been utilized to make methane. So this can be removed and discarded. There are three components. This is called digester. Okay. This is called the floating lid. Okay. Everyone is clear?
Yes, sir.
So we put cow dung plus water in the collector tank, we let it sit, methanogens increase, they go into this digester where lots of methane, hydrogen disulfide and CO2 is produced. All that gas is collected through a floating lid and can be used as a fuel source. And there is a sludge collector which is connected to this digester main tank, from where excess of the solution keeps getting collected and it can be removed. Okay. So in rural areas, it is still a very good and major source of fuel, where LPG connections, gas cylinders, inductions, microwaves, all these are not present. Is it clear, everyone?
Yes, sir.
Okay. Please draw this and let me know when you're done. Let me know when you're done, people. Shall I move forward?
One minute, sir.
Yep. So this diagram, remember this, it's simple. You just, according to your need, you have to make 3 big underground chambers, the one being the biggest, which is the digester, and the main chamber. From one side you will add, so that's the dung and water collector tank, and from the other side you will keep removing what is not useful anymore, which is the sludge collector, and then the sludge is removed. So it's a simple plan. Okay, shall I move, Safal? Are you still doing?
Yes, sir.
Okay, very well. So we studied microbes both in sewage treatment and biogas plant. Now I'm going to tell you a little bit of genetic engineering in biology, where we have utilized a microbe for it. So microbes in producing biocontrol agents. Now, what do we mean by biocontrol agents? Anyone? Split the term biocontrol agents. What is it? What does it sound like? Anyone? People? Biocontrol agent. No? There are certain chemicals that control many kind of life forms. Now, what kind of life forms do we control in our day-to-day life around us? We don't want them to be around us, or we don't want them to flourish. Any idea?
Mosquito.
Yes, mosquito being one. So what do we do to control mosquito population?
So we spray that chemical
You can spray a chemical, basically.
or put a mesh around the windows.
Yeah, putting a mesh is like where we are not allowed. So it's a simple physical barrier. I'm talking about more chemical, like some chemical or drugs or any other terms. We use these solutions, right? Vaporizing solutions, which are like all out, good night, all these things, correct? They
Yes.
kill the mosquito and not us in that concentration. Okay. Now, a biocontrol agent is one life form that help us to control the population of another life form. So we are not producing or involving chemicals here directly. We are involving microbes only. Now, in agriculture, what is the major problem that farmers face?
Pests.
Pests, right? Everyone agree?
Sir.
So major issue in agriculture is insects or pests. Now, what do they do? Why do we call it insects or pests? they are called insects and pests because they affect the crop. Simple. They kill the crop, eat the crop that we need for ourselves. They do not allow the crop to live long enough to produce viable seeds, etc. Whatever they do, they affect the crops and we don't like it. You know, we can also control the population of mosquitoes also naturally. So there are some natural predators of mosquitoes and they're called dragonflies. You know that? So what people do if a region is problematic, heavily, and where do these mosquito breed to begin with? Let me ask you that.
They're in water bodies.
What kind of any water, like in oceans also they can breed, like mosquitoes can breed in
The
oceans?
still water bodies.
Okay, still. So let's say still water body you're saying. So let's say any water body source which is very salty water source, but it is still, like Dead Sea. Can mosquitoes spread, or they can breed there? Mosquitoes need fresh water to breed. Okay, that's what I was asking. It cannot be marine or saline water, and it also cannot be very polluted water. So they have a preference towards clean water. So if at a place there is a, let's say, a lake which has a clean water, fresh water source, and you are seeing that there is lots and lots of mosquito breeding in that lake, so will you take all out and go and keep dumping all outs or oils in that lake?
No.
What will you do then? Think about it.
So we can release dragonfly.
You can start breeding dragonflies around that lake or in the water, because dragonflies also likes to live around water, and they are predators for the mosquito larvae, so they will keep the mosquito population in check. Correct? That is one way to use one life to regulate the other. I was just giving you an example with that. You understood?
Yes, sir.
Everyone? Okay, good. Now, how can we use microbes for producing a kind of a biocontrol agent? So in agriculture, insects and pests are major issues. We all agree, correct? So what happens is, it's figured out that there is a bacterial strain. Okay. There is a bacterial strain which is called Bacillus thuringiensis. Thuringiensis. Okay, it's often written as, it's called BT. Okay, now what does this BT does? It's very easy to get these bacterial spores or these bacteria as dry powders. So all you need to do is take that bacteria, farmers use it, and make a solution of that bacteria and spray this bacteria on the leaves of your plants or crops that you want to protect from a beetle or insects. Okay, so there are many beetles and insects that like to feed on the leaves or other parts of the agricultural plant itself, which will destroy the crop. So what they do is, instead, this Bacillus thuringiensis has been shown in nature that if an insect or a beetle, let's say, so it eats Bacillus thuringiensis. So if you spray it on the plant, then if these insects eat those bacterial spores or the bacteria, then what happens? when the insect lay eggs, okay, and become larvae come out of it. So in the gut of the larva, this bacteria goes into the gut of the insect larva and releases a toxin which kills the larva. Now, having said that, can you tell me how are we going to utilize it? Anyone? I told you the crux. Now tell me detailed. What can we do? Do you think there is something advantageous that we can use here, or no one thinks anything?
So if it kills the insects, we can keep the insects in check.
Sorry, come again.
So if we spray this in a plant, can we still eat the plant or be toxic for us also?
Yes, it can be toxic for us. So, but mostly in this case, we know that it is specifically toxic to the insect. So it kills the insect. So that's why we can use it. But do you think spraying the plant with this bacteria so that insects eat it up and this toxin is released and they die, do you think it's a good it's the ideal condition or the good idea, the best idea? Is there anything that we can do? No idea, people. So you have to participate in discussion, right? Okay, let me lead you. Someone has to answer. So I have Bacillus thuringiensis. If insects eat it, they'll produce a toxin in the gut. That toxin will lead to the perforation of the gut and will kill the insect. Now, what scientists have done it, they realize that it secretes, what it secretes in that insect's gut is called Bt toxin. Okay, so there's a gene for it, Bt toxin gene. Now, if I can put this gene only into the plant body, you understand? So genetically, I took that gene and put it in the plant body, expressed it in the plant. Do you think it's going to be it's going to do something? Do you understand? Can we do this or can we not?
Sir, can you repeat the question, please?
Yes, the question is, we know that there's a bacteria called Bacillus thuringiensis that is spread over the plants that we want to protect. Now, when we spray that and then other kind of insects that feed on these plants come and start eating them. So they're also eating this bacteria. And when they lay eggs, these insects, and then the larval, the bacteria produces toxin called Bt toxin that perforates the gut of the insect and kills the insect. So this is what people have been doing. Now with better scientific techniques, what we can also do is that we can take that Bt gene, the Bt toxin gene from the bacteria, and we can put it in the plant cell or all the cells, like in the plant zygote, so that all the plant cells have this Bt toxin gene. Do you think it serves any purpose? It has any advantage?
So then we don't have to spray it on the plant. They will
First,
naturally.
perfect. First, we don't have to spray it, but how do you think this is going to work? What's going to happen? But the plant just have the gene, right?
So since it has the genes, it will automatically release the chemical in its body.
Perfect. Since plant only now have the gene of Bt toxin, inside the plant cell, inside the plant tissue, there will be this protein or this toxin formed, right?
Yes, sir.
Now, even if we do not spray this bacteria, and even if when these insects, which normally eat these plants, will come and eat the plant tissues, this toxin will go into their gut and
Yes.
it will perforate their gut and it will kill these pests.
Yes, sir.
So do you see what we did there? We created plants which are resistant to attack by insect pests. And one such example you can write down by expressing yes, Vafia, do you have a question?
No, sir.
Okay. By expressing the Bt toxin gene in the desired plant, one can make resistant plants, or one can make plants that are resistant to attack by insect pests. Make sense, everyone? Make sense?
Yes, sir.
Yes. So now, one example and a famous one which can be asked in your entrance and also in your exams is Bt cotton. So it is a GMO, genetically modified organism, or GMC, genetically modified crop. Bt cotton is a genetically modified crop, GMC. Okay. So about this Bt cotton's production, we'll study about in the biotechnology chapter. It is a part of your biotechnology syllabus. But just know that what we used here, basically, we used bacteria called Bacillus thuringiensis. First, we were using it physically to spray, and with advanced genetic concepts, we can just make genetically modified cotton, Bt cotton. Now, if these insects, if these beetles come and start eating up this cotton plant, these toxins will be formed in their gut, will reach their gut and will perforate the whole gut and will kill the pathogen. Okay. Similarly, yes. So this is one example of Bacillus thuringiensis. You can write here as one. Similarly, there is another group of bacteria found which are called baculovirus. Have you heard of baculovirus ever? Baculoviruses. Heard of it? No. Okay. So I
No.
want you to understand. Okay. All of you have NCERT with you?
Yes, sir.
Okay. So please open your NCERT and come to page 187. And this homework is for Minashi. Minashi, are you there? Can you hear me?
Yes, sir.
Okay, cool. So others also will read, but Minashi will answer. I'm giving you 3 minutes to read the last paragraph on page 187 before microbes as biofertilizers. Do you see microbes as biofertilizers written? Before that, there is a paragraph talking about baculovirus as pathogen. And before that, there is also one about trichoderma. So the two paragraphs. A biological control being developed for the use of treatment of plant disease is the fungus, trichoderma, from there. So you have to read these two paragraphs and tell me what you understood from the paragraph. I'm giving you 3 minutes. Okay, Minashi?
Okay.
I can also ask others for clarification. So please read it. Your time starts now. I hope everyone is reading. Is it easy or is it difficult? Take a couple of more minutes, try to understand it, and then I'll ask you to explain. Okay, time is up. Minashi, do you want more time?
It's fine, sir.
Okay, so can you start explaining? So before let's first start with baculovirus because we have written it already. What is baculovirus?
Baculovirus are the pathogens that attack the insects, and the genes that is present in this virus is nucleopolyheteroviruses, and these
No, that's not the
pathogens
genes. Read carefully. What is it written?
biological agents are in the genus.
Yes. So these viruses, the baculoviruses, are the pathogens that attacked insects. Okay, so these pathogens are not good for insects. They kill insects, and we want to get rid of insects. So we can use this baculovirus as a biological control agent because they are in the genus. So baculovirus belongs to the genus, right? These are the species. And to which genus do they belong to? Minashi?
Nucleopolyheterovirus.
Yes. What? Nucleo
Polyheterovirus.
polyhedrovirus.
Sure.
Nucleo Hydrovirus. So this is the genus. Okay. And what apart from that? Yes. So they
Then,
okay, they attack the insect, right?
yes.
They attack and kill. Yes, next.
Then these viruses do not have any side effects on plants, mammals, or birds, but like it only kills the insects that are targeted.
Yes, now that's the most important property by due to which we can, because someone asked, can these bacteria affect or can this toxin affect humans, right? If we eat that plant. So in cases where the one biological organism affects the other, but can also affect more than the others. Like if it, let's say this toxin was affecting insects and was also affecting humans, could we have used it as a biocontrol agent then?
No.
No, right? Because we need something which specifically kills the insect, but even if we eat it, it is not harmful to us. So these baculoviruses are very, very specific. So they are species specific. Okay. Species specific and a very narrow range. Only a few insects they will give. Not narrow range. Insecticidal impact. Narrow range. Insecticidal impact. This is a very important property. Yes, Minashi, continue.
The last point I didn't get.
Sorry?
The last point I didn't get. I mean, this.
Yes, what is the last point?
This is especially desirable when beneficial insects are being conserved to aid in overall integrated pest management or when an ecologically sensitive area is being treated.
Yes. So the last point is related to the second last point. Will you read the second last point also? The second last point of the same paragraph, Minashi. Are you there, Minashi?
Sorry. I forgot to unmute my mic.
Sorry?
I
Okay.
forgot to
Oh,
unmute my-
okay. And you're speaking. Okay.
Yeah.
That's okay. Yes,
They
come on.
have been shown to have no negative impacts on plants, mammals, birds, fish, or even non-targeted insects.
Yes. So you understand this word non-targeted insects? So this is very narrow range. Baculovirus only kills particular insects. So even in the insect phylum, it will not end up killing or it will not be harmful for all the insects. Now this is this is what the last point says. This is specially desirable when beneficial insects are being conserved. So suppose you want to kill some kind of insects that are destroying your crops by using baculovirus. But you also do not want all the insects to die because there are good insects also. Insects are pollinators, correct? They are very important pollinators. For example, bees. Let's say I want to kill the beetles that eat my plant, my crop. But I don't want to kill honeybees because they are the ones which are pollinating my crop. So one insect is harming the crop, another insect is benefiting the crop, pollinating the crop. So that is what this technique says, that baculovirus are so specific to certain insects that they can be used as a good candidate to work or to conserve particular type of insect species while eliminating the other. Does it make sense, everyone? People, does it make sense to you?
Yes, sir.
Yeah. Okay. So, and what about, so this was baculovirus, okay? So they help in ecologically sensitive areas where we want to conserve certain insects which are helpful to the plant, and we also simultaneously wants to eliminate certain insects which are not good to the plant. Make sense? So if this question comes, what kind of microbes can be used in an insect conservation program where we also want to get rid of some other insects, then we can use the baculovirus. Okay. Okay. Yeah. Okay. Brilliant. So now we have the last part for this chapter, which we will finish today, and then I'll be starting with biotechnology in the next class. So this last part is microbes as biofertilizers. Now it's along the same lines as I have told you above for BT cotton, but here, how are we using it as biofertilizers and which are used? So again, let me go this time to Samiha. Samiha, are you there? Can you speak up?
Yes, sir.
Yes. So let's do this in class activity quickly. So you will read this microbes as biofertilizers paragraph. Okay. So there are, I can see 3 paragraphs. Okay. So
Yes, sir.
first paragraph you will read, Samiha. Second, Mariam. Mariam, are you there? Can you speak? Can you participate in the class, Mariam? Okay, Mariam is not responding. Saifullah and Wafia. I need 3 people.
Yes, sir.
Okay, so Saifullah will read the second paragraph, which is about the fungi. Fungi are also known to form symbiotic associations. Before that, it will be Samiha, then fungi paragraph will be Saifullah, and the last cyanobacteria paragraph, it will be Wafia. Wafia, can you do that?
Yes, sir.
Okay, cool. So, but you have to read pura. It's not like you can just read your paragraph and understand. The context is in the above. You have to just explain that. So, how much time do you need to read these 3 paragraphs? 5 minutes will be okay. Okay, let's say 6. These are not very big paragraphs. So 6 minutes. Okay. Samiha, Saifullah, and Wafia. Okay. Yes, no.
Okay, sir.
Okay, so your time starts now, and you can start reading. And I'm here only. If you find any difficulty, just tell me. So I want to see how well you understand the text from NCERT. Your time starts now. Okay, I'll be silent now. And remember, take it seriously. It's an in-class evaluation, kind of. Any difficulty, people?
No, sir.
No, sir.
Okay, Wafia?
No, sir.
Are you done? Can we begin or you are finishing up? Technically, you have a couple of minutes more. Okay, so we'll begin with Samiha, right? Are you there, Samiha?
Yes, sir.
Shall we begin?
Yes.
Okay, so this part is microbes as biofertilizers, right? So let me write it down and then we'll start with Samia. What does she understands? Microbes as biofertilizers. Yes, Samia. Go on.
Um.
So what does
So
the first paragraph tells? Yes.
that the environmental pollution
Mhm.
is a big concern
Mhm.
in today's world,
Yes.
and one of the reasons is the use of chemical fertilizers, which has been in use for a long time. But we have
Why do we need fertilizers, Samia? Let me just ask you. Why do you think we need
to
fertilizers?
enrich the soil.
Right. And why is this pressure on farmers to use fertilizers and manures is increasing?
It's because fertilizers help the crop to grow faster, and it
Why do we want crops to grow faster? What is this pressure of growing crops faster in more numbers? Why?
so they can sell it more and get more money.
Why do we want to sell it more? Why do we want more money? There's one simple answer. There are more mouths to feed on the planet than we can generate. Simple, right? Population
Yes.
explosion of humans is the main reason that we need more agricultural land. And if we cannot get more agricultural lands, if we can, then we are burning forests and removing forests and making it fields. But if we can't, then we need to take more. We need to get more out of the same piece of land. That's where this drive and this pressure of using fertilizers, biofertilizers, switching to organic farming because of deteriorating quality and health, all this thing is coming. Yes, you continue. Sorry.
Yeah, so we have realized how dangerous the chemical fertilizers are. So nowadays, people are switching to organic farming, which is using minimal fertilizers, and we use mostly biofertilizers for this. And in this paragraph, we have been given two such biofertilizer sources, bacterias, which is one of them is rhizobium bacteria, which lives symbiotically with the roots of leguminous plants.
Correct. What are
Um,
you have any idea?
yeah, so what rhizobium does is that it converts the atmospheric nitrogen into the usable nitrogen as a nutrient for the plants.
Yeah, in the form of nitrites and nitrates. So plants take up nitrogen, they cannot take it in the gaseous form. So it needs to be fixed in the form of nitrates and nitrites. That's it does. But I was asking, what is leguminous plants?
Um,
Leguminous plants are your dal and pulses, beans,
yes.
legumes. Yes, okay. These are legumes with high protein content in them. And a very important component of protein is nitrogen. So nitrogen is required to make protein. Okay.
Yes, sir.
Okay. Very well.
Another example we have of bacteria which do not need symbiotic associations and they can live freely in the soil. They are Azospirillum and Azobacter. These
Azotobacter.
azoto-
Azospirillum and Azotobacter. Yep.
they can just freely live in the soil and they can fix the atmospheric nitrogen and just enrich the nitrogen content of the soil.
Of the soil, and from the soil then plants can directly take it. So one is a symbiote, is rhizobium, and two are free living, but they are doing basically the same thing, correct?
Yes,
And
sir.
this kind of activity helps us to improve the plant yield, correct? So
Yes.
these three bacteria are an example of microbes as biofertilizers, right? Because they are increasing the yield. Is this what is your home message from this paragraph, Samia? Yes.
Yes, sir.
Yeah. Okay. Very good. Very well explained. Well done. Okay. Next. Who's next? Saifullah. What does the second paragraph tells us about?
So says about the symbiotic association with fungi and plants.
Okay. So the first paragraph, Samia talked about bacteria. Now, Saifullah will talk about fungi. Very good.
So the fungi are known to form a symbiotic association with plants. This association is known as mycorrhiza.
Mycorrhiza. Yes.
Yes. Um, the genus Glomerus is
That you don't have to remember. Okay. That just know that there's a genus. That will not be asked. Okay. But what is the take home message from this short paragraph? How is it helping the plant?
so the fungal part of this association, like they absorb the phosphorus from the plants and pass it to the plant.
Yes. So the fungi is providing plants with another important nutrient. So in the first paragraph, Samiha told us that these bacteria, they were providing plant with an important component called nitrogen for the leguminous plants to make protein. Here you are saying that there is a fungal symbiont as well, which provides the plant with phosphorus, right? Any
Yes.
other benefit?
So the plants having this association is resistant to root-borne pathogens, tolerance to salinity and drought.
So which means that this association is very good for plants. They can tolerate salinity better, they can tolerate less water conditions, and they are also resistant to many pathogens that attack on the root, correct? And plus they get phosphorus. That's the main benefit.
Yes, sir.
Yes. Okay, very good. So I hope everyone is writing, because I'm not writing and your own peers are dictating things to you. So these kind of activities we will keep doing as in class evaluation. Well done. Thank you so much, Saifullah. So let's go to the last part, last paragraph, and also the last part of this chapter. Let's go to Wafia.
So last about is cyanobacteria. Cyanobacteria they are an autotrophic microbes, and they can be found in aquatic and terrestrial environments. And like the rhizobium bacteria, cyanobacteria they can also fix the atmospheric nitrogen. And examples of cyanobacteria are Anabena, Nostoc, and Oscillatoria.
So
And
good. There is one term which I was curious about. There's something written as blue-green algae. What is blue-green algae?
blue-green algae, they also are organic matter, and they increase the soil fertility.
Blue-green algae is another name for cyanobacteria. Okay, this is where they trick you in entrances. Blue-green algae sounds like that this organism is an algae, but remember, blue-green algae is a bacteria. Write it down. Write it down right there in your books or in your copies. Blue-green algae, also known as BGA, is not an algae, but it is a bacteria. Okay. Written, everyone? Is the another name for cyanobacteria. It's a autotrophic bacteria. Yes, continue, Wafia. Let's finish.
So in the paddy fields and all, the cyanobacteria, they are like important biofertilizers.
Mhm.
And now in currently we can find the biofertilizers in the market commercially, and nowadays the farmers are mostly using the biofertilizers to replenish the soil nutrients, and they reduce the dependence on the chemical fertilizers.
Yes, because chemical fertilizers in a long run are not very good either for the plant, for us who are eating those plant source food, and also for the soil. Okay, so shifting to microbes as biofertilizers is a good option, but the only thing is the availability, the feasibility. Everyone cannot get it in the amount that is required. But yes, this was for you to study, and this marks the end of this chapter. Okay, please go back, revise about all the microbes that we have studied, which are good for human. And in the next class, I'll start with biotechnology.